Skeletal muscle. The best known feature of skeletal muscle is its ability to contract and cause movement. Skeletal muscles act not only to produce movement but also to stop movement, such as resisting gravity to maintain posture. Small, constant adjustments of the skeletal muscles are needed to hold a body upright or balanced in any position. Muscles also prevent excess movement of the bones and joints, maintaining skeletal stability and preventing skeletal structure damage or deformation. Joints can become misaligned or dislocated entirely by pulling on the associated bones. Muscles work to keep joints stable. Skeletal muscles are located throughout the body at the openings of internal tracts to control the movement of various substances. These muscles allow functions, such as swallowing, urination, and defecation, to be under voluntary control. Skeletal muscles also protect internal organs, particularly abdominal and pelvic organs, by acting as an external barrier or shield to external trauma and by supporting the weight of the organs. Skeletal muscles contribute to the maintenance of homeostasis in the body by generating heat. Muscle contraction requires energy, and when ATP is broken down, heat is produced. This heat is very noticeable during exercise, when sustained muscle movement causes body temperature to rise, and in cases of extreme cold, when shivering produces random skeletal muscle contractions to generate heat. Each skeletal muscle is an organ that consists of various integrated tissues. These tissues include the skeletal muscle fibers, blood vessels, nerve fibers, and connective tissue. Each skeletal muscle has 3 layers of connective tissue, called mysia, that enclose it and provide structure to the muscle as a whole, and also compartmentalize the muscle fibers within the muscle. Figure 1. Each muscle is wrapped in a sheath of dense, irregular connective tissue called the epimysium, which allows a muscle to contract and move powerfully while maintaining its structural integrity. The epimysium also separates muscle from other tissues and organs in the area, allowing the muscle to move independently. Inside each skeletal muscle, muscle fibers are organized into individual bundles, each called a fascicle, by a middle layer of connective tissue called the perimysium. This fascicular organization is common in muscles of the limbs. It allows the nervous system to trigger a specific movement of a muscle by activating a subset of muscle fibers within a bundle, or fascicle of the muscle. Inside each fascicle, each muscle fiber is encased in a thin connective tissue layer of collagen and reticular fibers, called the endomysium. The endomysium contains the extracellular fluid and nutrients to support the muscle fiber. These nutrients are supplied via blood to the muscle tissue. In skeletal muscles that work with tendons to pull on bones, the collagen in the 3 tissue layers, the mysia, intertwines with the collagen of a tendon. At the other end of the tendon, it fuses with the periosteum coating the bone. The tension created by contraction of the muscle fibers is then transferred through the mysia to the tendon, and then to the periosteum to pull on the bone for movement of the skeleton. In other places, the mysia may fuse with a broad, tendon-like sheet called an aponeuroses, or to fascia, the connective tissue between skin and bones. The broad sheet of connective tissue in the lower back that the latissimus dorsi muscles, the lats, fuse into is an example of an aponeuroses. Every skeletal muscle is also richly supplied by blood vessels for nourishment, oxygen delivery, and waste removal. In addition, every muscle fiber in a skeletal muscle is supplied by the axon branch of a somatic motor neuron, which signals the fiber to contract. Unlike cardiac and smooth muscle, the only way to functionally contract a skeletal muscle is through signaling from the nervous system. Skeletal muscle fibers. Because skeletal muscle cells are long and cylindrical, they are commonly referred to as muscle fibers. Skeletal muscle fibers can be quite large for human cells, with diameters up to 100 μm and lengths up to 30 cm. 11.8 in the sartorius of the upper leg. During early development, embryonic myoblasts, each with its own nucleus, fuse with up to hundreds of other myoblasts to form the multinucleated skeletal muscle fibers. Multiple nuclei mean multiple copies of genes, permitting the production of the large amounts of proteins and enzymes needed for muscle contraction. Some other terminology associated with muscle fibers is rooted in the Greek sarco, which means flesh. The plasma membrane of muscle fibers is called the sarcolemma. The cytoplasm is referred to as sarcoplasm, and the specialized smooth endoplasmic reticulum, which stores, releases, and retrieves calcium ions, Ca++, is called the sarcoplasmic reticulum. Sr. Single quote. Figure 2. As will soon be described, the functional unit of a skeletal muscle fiber is the sarcomere, a highly organized arrangement of the contractile myofilaments, actin, thin filament, and myosin, thick filament, along with other support proteins. The sarcomere. The striated appearance of skeletal muscle fibers is due to the arrangement of the myofilaments of actin and myosin in sequential order from one end of the muscle fiber to the other. Each packet of these microfilaments and their regulatory proteins, troponin and tropomyosin, along with other proteins, is called a sarcomere. The sarcomere is the functional unit of the muscle fiber. The sarcomere itself is bundled within the myofibril that runs the entire length of the muscle fiber and attaches to the sarcolemma at its end. As myofibrils contract, the entire muscle cell contracts. Because myofibrils are only approximately 1.2 feet μm in diameter, hundreds to thousands, each with thousands of sarcomeres, can be found inside one muscle fiber. Each sarcomere is approximately 2 μm in length with a three-dimensional cylinder-like arrangement and is bordered by structures called Z-discs, also called Z-lines, because pictures are two-dimensional, to which the actin myofilaments are anchored. Figure 3. Because the actin and its troponin-tropomyosin complex, projecting from the Z-discs toward the center of the sarcomere, form strands that are thinner than the myosin, it is called the thin filament of the sarcomere. Likewise, because the myosin strands and their multiple heads, projecting from the center of the sarcomere toward but not all the way to the Z-discs, have more mass and are thicker, they are called the thick filament of the sarcomere. The neuromuscular junction. Another specialization of the skeletal muscle is the site where a motor neuron's terminal meets the muscle fiber, called the neuromuscular junction. NMJ. Single quote. This is where the muscle fiber first responds to signaling by the motor neuron. Every skeletal muscle fiber in every skeletal muscle is innervated by a motor neuron at the NMJ. Excitation signals from the neuron are the only way to functionally activate the fiber to contract. Excitation-contraction coupling. All living cells have membrane potentials, or electrical gradients across their membranes. The inside of the membrane is usually around -60 to -90 millivolts. Relative to the outside, this is referred to as a cell's membrane potential. Neurons and muscle cells can use their membrane potentials to generate electrical signals. They do this by controlling the movement of charged particles, called ions, across their membranes to create electrical currents. This is achieved by opening and closing specialized proteins in the membrane called ion channels. Although the currents generated by ions moving through these channel proteins are very small, they form the basis of both neural signaling and muscle contraction. Both neurons and skeletal muscle cells are electrically excitable, meaning that they are able to generate action potentials. An action potential is a special type of electrical signal that can travel along a cell membrane as a wave. This allows a signal to be transmitted quickly and faithfully over long distances. Although the term excitation-contraction coupling confuses or scares some students, it comes down to this. For a skeletal muscle fiber to contract, its membrane must first be excited. In other words, it must be stimulated to fire an action potential. The muscle fiber action potential, which sweeps along the sarcolemma as a wave, is coupled to the actual contraction through the release of calcium ions, Ca++, from the SR. Once released, the Ca++ interacts with the shielding proteins, forcing them to move aside so that the actin binding sites are available for attachment by myosin heads. The myosin then pulls the actin filaments toward the center, shortening the muscle fiber. In skeletal muscle, this sequence begins with signals from the somatic motor division of the nervous system. In other words, the excitation step in skeletal muscles is always triggered by signaling from the nervous system. Figure 4. The motor neurons that tell the skeletal muscle fibers to contract originate in the spinal cord, with a smaller number located in the brainstem for activation of skeletal muscles of the face, head, and neck. These neurons have long processes, called axons, which are specialized to transmit action potentials long distances. In this case, all the way from the spinal cord to the muscle itself, which may be up to 3 feet away. The axons of multiple neurons bundle together to form nerves, like wires bundled together in a cable. Signaling begins when a neuronal action potential travels along the axon of a motor neuron, and then along the individual branches to terminate at the NMJ. At the NMJ, the axon terminal releases a chemical messenger, or neurotransmitter, called acetylcholine, ACH. The ACH molecules diffuse across a minute space called the synaptic cleft, and bind to ACH receptors located within the motor endplate of the sarcolemma on the other side of the synapse. Once ACH binds, a channel in the ACH receptor opens and positively charged ions can pass through into the muscle fiber, causing it to depolarize, meaning that the membrane potential of the muscle fiber becomes less negative, closer to zero, as the membrane depolarizes. Another set of ion channels called voltage-gated sodium channels are triggered to open. Sodium ions enter the muscle fiber, and an action potential rapidly spreads, or fires, along the entire membrane to initiate excitation-contraction coupling. Things happen very quickly in the world of excitable membranes. Just think about how quickly you can snap your fingers as soon as you decide to do it. Immediately following depolarization of the membrane, it repolarizes, re-establishing the negative membrane potential. Meanwhile, the ACH in the synaptic cleft is degraded by the enzyme acetylcholinesterase, ACH, so that the ACH cannot rebind to a receptor and reopen its channel, which would cause unwanted extended muscle excitation and contraction. Propagation of an action potential along the sarcolemma is the excitation portion of excitation-contraction coupling. Recall that this excitation actually triggers the release of calcium ions, Ca++, from its storage in the cell's SR. For the action potential to reach the membrane of the SR, there are periodic invaginations in the sarcolemma, called T-tubules. T stands for transverse. You will recall that the diameter of a muscle fiber can be up to 100 μm. So these T-tubules ensure that the membrane can get close to the SR in the sarcoplasm. The arrangement of a T-tubule with the membranes of SR on either side is called a triad. Figure 5. The triad surrounds the cylindrical structure called a myofibril, which contains actin and myosin. The T-tubules carry the action potential into the interior of the cell, which triggers the opening of calcium channels in the membrane of the adjacent SR, causing Ca++ to diffuse out of the SR and into the sarcoplasm. It is the arrival of Ca++ in the sarcoplasm that initiates contraction of the muscle fiber by its contractile units, or sarcomeres. Chapter Review. Skeletal muscles contain connective tissue, blood vessels, and nerves. There are 3 layers of connective tissue: epimysium, perimysium, and endomysium. Skeletal muscle fibers are organized into groups called fascicles. Blood vessels and nerves enter the connective tissue and branch in the cell. Muscles attach to bones directly or through tendons or aponeuroses. Skeletal muscles maintain posture, stabilize bones and joints, control internal movement, and generate heat. Skeletal muscle fibers are long, multinucleated cells. The membrane of the cell is the sarcolemma. The cytoplasm of the cell is the sarcoplasm. The sarcoplasmic reticulum, SR, is a form of endoplasmic reticulum. Muscle fibers are composed of myofibrils. The striations are created by the organization of actin and myosin, resulting in the banding pattern of myofibrils. Make sure to smash the like and subscribe buttons to receive notifications all new contents.